Hello, everybody, welcome to watch Creative Biolabs video about immune system and immune therapy. Today, we are going to share some basic knowledge about T-Cell Receptor Engineered T-Cell technology and its application on tumor therapy. Immunotherapy is becoming part of the systemic treatment options for cancer patients. In particular, immunomodulating agents, like the checkpoint blocking antibodies such as PD-1 and CTLA-4 monoclonal antibody that block the negative regulators of T-cell immunity. In addition to immunomodulating drugs, another promising immunotherapeutic approach is adoptive T-cell therapy, which is an immunotherapeutic treatment that based on the principle of transferring autologous or ex vivo expanded tumor-specific T-cells to patients. There are different strategies of ACT. Tumor-infiltrating lymphocytes, cytokine-induced killer cell, chimeric antigen receptor T-cell which we have reviewed in another video, and TCR engineered T-cell which we will talk about in this video are all be considered and developed. To better understand how TCR engineered T-cell technology works, we should first know about the TCR structure. TCR is not just a single protein but a protein complex consists of a group of membrane proteins including TCR alpha and beta as antigen recognition domain, CD3 gamma, epsilon, delta and zeta as signal transduction domain. The TCR alpha and beta domain are translated by spliced TCR alpha and beta mRNA which derives from the germline TCR alpha and beta gene respectively, and form a heterodimer. The anti-tumor specificity of T-cell is mainly determined by this heterodimer receptor. TCR-T is such a kind of T-cell, which receptor genes are genetically engineered to obtain higher anti-tumor affinity and tumor-killing efficacy. Generally, there are 5 steps to develop TCR-engineered T-cells. First of all, we must choose a proper antigen as the target of TCR-T cell. These antigens could be expressed tumor-associated antigen, or tumor-specific endogenous proteins. Then, high-affinity T-cell clone must be isolated, and TCR alpha and beta gene should be cloned as well. The next step is to package the specific TCR gene into vectors, such as retroviral or lentiviral vector to deliver TCR gene into donor T-cells, which are isolated from the patient's peripheral blood. After successfully gene transfer, the transgenic T-cell should be further processed to improve the TCR performance. When all of the preclinical tests are done, the TCR T-cell could finally adoptive transfer into patient's body. However, high-affinity T-cell receptors are not so easy to obtain. Autologous tumor-infiltrating lymphocytes were the first to be used as high-affinity TCR sources for its natural anti-tumor specificity. Although the results are promising, the majority of patients are not eligible for TIL therapy, as not all patients have pre-existing tumor-reactive T-cells, and in many cancers it is still difficult to identify and obtain tumor-reactive lymphocytes. Several creative approaches have been used to generate TCR that are specific for other antigens that are expressed by a wider variety of tumor cell types. Such approaches include using allogeneic T-cells from individuals who are not tolerant of patient TIAs, or using those from transgenic mice that express human MHC. In these approaches, T-cells are incubated with tumor cells from the patient, and TCR genes are cloned from T-cells that react to the tumor cells. Alternatively, a DNA library of many millions of TCR genes can be generated in bacteriophages, and tumor-reactive TCR genes can be obtained from phage display screening. Tumor antigen-specific TCR needs to be genetically transfected into the genome of T-cells and stably expressed. To assure co-expression of both chains, the TCR alpha and beta genes are most commonly linked via picornavars to a ribosomal skip peptide. For human applications, gene transfer platforms such as gamma-retroviral, lentiviral vectors, or transposons that can mediate stable gene transfer are the systems of choice. The 2 virus-based systems are complex biological reagents that require extensive safety testing for human applications, but they mediate very high gene transfer efficiencies, and have been used for over 2 decades in human studies. Transposons are a relative newcomer in the human gene therapy field, and have the advantage that they are plasmid DNA-based, are much simpler to produce, and require less upfront safety testing. Unlike antibodies, the affinity of many naturally occurring TCRs for their target peptide is low. Therefore, steps to improve the performance of TCRs through protein engineering should be made. These include strategies to improve TCR affinity, increase cell surface expression, and prevent mixed dimer formation between the introduced and endogenous TCR chains. Such mixed dimers would not target the tumor antigen. Single and dual amino acid substitutions in TCR-CDRs can enhance antigen-specific T-cell functions. Development of hybrid TCRs, in which the human constant region is replaced by a murine constant region, has been shown to improve specific chain pairing and TCR expression. Introduction of an additional cysteine bridge or a second disulfide bond in the constant region of the TCR alpha and beta chains also improves pairing. The first reported clinical studies using TCR gene-modified T cells in the cancer setting involved the use of human T cells, transduced with an anti-MART1 TCR transgene that was isolated from TILS from patients with melanoma. Recently, several clinical trials have been reported, documenting the clinical efficacy of gene-modified T cells for treatment of other cancers. These trials use TCR engineered T cells and have shown clinical benefit in several different cancers, including melanoma, colorectal cancer, synovial cell cancer, neuroblastoma, and lymphoma. The main advantage of using TCRs to target tumors is that they function through well-understood T-cell signaling pathways and are the natural means by which the body clears foreign elements. The main disadvantage of TCR-based anti-cancer therapies is that the biology of the TCR restricts it to an HLA type, and alpha/beta TCRs cannot target non-protein tumor antigens such as carbohydrate or lipid antigens. Altering the specificity of T-cell receptor is one of the popular strategies to genetically modify T cells to enhance the tumor-killing activity of T cells. With high-affinity TCR technology, Creative Biolabs is capable of identifying the candidate target tumor antigen and the sequence of tumor-specific TCR to create tumor-specific T cells. We have high-qualified groups and advanced technologies to perform TCR engineering services covering every unit of manufacturing and delivery pipeline. From a tumor-reactive T-cell or active anti-tumor T-cell antigens, the appropriate target sequence is introduced to modify T-cells to target a broad range of tumors with improved specificity. With engineering TCR construction and a series of assays, such as transgene expression, TCR CD3 stability, lymphocyte antigen reactivity, cytotoxicity, and animal experiments. Thank you for watching our video. If you have any questions or need more detailed information about TCR technology, please do not hesitate to contact us by email or visit our website. We are more than happy to help you with your TCR project.